Thank you very much for having me. Same as Dr. Ake, I was in Europe last week. I got back yesterday, so if I drop the microphone, I'll pick it up. We'll keep going, but I'll try to coordinate the two here smoothly. So I guess we're gonna go back to the bench side now. I'm gonna sort of hit on a lot of different applications of the technology we have developed. So I'll be more than glad to talk to anybody that have more questions during the break or tonight. So I'll be talking to a technology that we developed, which is magnetic 3D bioprinting. And it's a technology that has several applications. So I'm gonna try to touch as many as I can. Quick, who we are. I think we all come, the first three talks came from the Gulf of Mexico. I'm also in Houston, so we're all in close by. And this is a different angle from it's a night view of the medical center. Dr. K showed the day view, so we're fairly close. And in 3D, it started from a collaboration between MD Anderson, I was at MD Anderson, and Rice University, which is right in the Houston Medical Center. And we developed the technology, we spin off into a company, and as of April, Griner Bio1, and that's why I have it there, acquired the technology. So there's various applications from basic research, very new, cool things that we're developing, to applications that we're selling. So as Dr. Ake mentioned, why 3D cell culturing, why we're talking about 3D bioprinting. I'll go quick through this. We all know, as we talked about earlier today, drugs cost a lot of money to develop. And part of the reason they cost a lot of money is because of the development process. A lot of drugs in the very early beginning, in the beginning of drug development, they're tested in monolayer cultures. And since we have a 3D bioprinting, I think we all believe that monolayer cultures are not really good representation of in vivo biology. So, and then you go to animals, which is, it's not human, so it's animal. It's usually rat physiology. Animal physiology is not the same physiology as human. So when the drugs get to humans, there's a high rate of drug failure from toxicity or poor efficacy. So the 3D world is to bridge the in vitro to human and hopefully reduce the cost of drugs or developing drugs. So how do we do magnetic 3D cell culture? As the name says, since we use magnetic fields, cells are now magnetic, we have to first magnetize the cells. So we call what we call the nanoshuttle. Nanoshuttle is a magnetic nanoparticle assembly of gold iron oxide polylysine. The nanoparticles just stick to the cells, electrostatic. It's known specifically via polylysine. The workflow is very similar to traditional 2D monolayer culture. You add your nanoparticles, they stick to the cells, you detach your cells. Same workflow as you use with monolayer culture, with 2D culture. And we developed two approaches. The first one actually was what we call magnetic levitation. So we magnetize the cells, and then we apply the magnetic field on top. And the cells lift off the bottom, and now they have the three-dimensional environment to develop the 3D structures. And then we went a step beyond, and we developed the bioprinting. The bioprinting, instead of having the magnet on top, we have the magnet on the bottom. So we can have different magnet shapes, position the magnets in different places, and we can actually print the cells. And we always use a cell-repellent surface, which means that the cells do not stick to the bottom. So because if you print the cells, on a surface that is not cell repellent, the cells will stick to it and grow as a monolayer. So, and generally speaking, these are what we call low throughput applications, which is 624 wells. And the high throughput, as you see, you can go all the way from 6 to 1,536 wells. So it's a really scalable technology. And that's one of the challenges in the field in general. When you go from a low throughput, large scale tissue structure it's very hard to use that same technique when you go to the high throughput, to very small wells. So there's, we really try to maintain the same technique throughout scales and throughput. So, but the key for our technique to work, it's not the magnetic field and it's not the nanoparticles. It's, I have to go here, it's cell-cell interaction. So you magnetize the cells, and this is what happens in the first 10 minutes of culture. You're just promoting cell-cell interaction. You just bring the cells together. So when you bring the cells together, they start excreting their own extracellular matrix and behaving like they do in vivo environment. And because we can apply the magnetic field and bring the cells together fast, we can generate spheroids within 24 hours. We can bring different cell types together. And as you have here, here's a GFP label glioblastoma, astrocyte label. We cultured them separately, and we brought them together. And you can actually see the glioblastoma invading the astrocytes here. We published this a few years ago in Nature Nanotech. Actually, this was the first publication we had on the technique when we came out with the technique. This is actually a diposphere, is a co-culture of pre-adipocyte stem cells that we co-culture with endothelial cells. And we differentiate the pre-adipocyte stem cells into adipocytes. The red here is pyrrolipin, which is a marker for adipocyte. The green are the endothelial cells that you can actually see sprouting into the tissue. And this was the cover tissue engineer a few years ago. But one thing that is important here, for us to generate this adiposphere model, which that has the endothelial cell phenotype within the structure, it wasn't just mixing the two together and voila, we got it. Because one thing that I always say, it's a disclaimer for me, I'm not a biologist, I'm a physical chemist. So when we developed this technique, this was very early on, we were working with this biology lab, and they came to me and it was like, okay, we want to do a co-culture of the pre-adipocyte stem cells and the endothelial cells. What cell ratio do we use? I look at them, it was like 50-50. And they're like, okay, we start on that. It did not work. 50-50 did not work. So we had to optimize the ratio of cells. And for us to get this sprouting, the phenotype, the organization, we had to go 95% the pre-adipocyte stem cells, 5% of the endothelial cells. Then you get this beautiful sprouting. So you really have to play with the microenvironment to get the results you want. A lot of times we focus in exactly positioning the cells, especially the bioprinting, but positioning can help. But I think the ratio and the biological conditions are more important because cells are smart. They go where they're supposed to go. So this is, I showed this for a few different reasons. It's a new technique. So from if I put in my marketing salesperson hat, in our world, the best marketing tool is a good publication. So we worked really hard to publish in good articles. And as is for students usually say, oh, I don't want to write papers, I don't want to write proposals, I'll go to industry. And it's not going to work. You're going to have to write papers, and if you go to a small company, you're going to have to write proposals as well. So, but we worked really hard to publish in good journals. And so it's a new technique, but we also use the nanoparticles. I'm sure you're thinking, okay, what did the nanoparticles do to the cells? They all ask. And over we have shown that the nanoparticles at the concentrations we use is just bioinert. They don't do anything to the cells. And we have a white paper that we compiled all the published data. We've looked at viability, proliferation, with and without the nanoparticles, protein expression, stem cell differentiation, inflammatory response with human primary cells, oxidative stress, gene expression, comparing to in vivo with and without nanoparticles. It's always the same. Those response, so we have published over and over that the nanoparticles don't do anything. And a lot of times when people really insist on, or are very skeptical of the nanoparticles, I ask, do we use serum in our cells? And people usually do. And your answer is, I'm not sure what's in serum, or especially from batch to batch. So at least here, you're usually getting the same thing over and over. So, okay, into the bioprinting. So the technique is very simple, as I said. This is what it is. And we have on the table there, you guys can stop by to see. It's a magnet array, See the pins in a cell repellent surface. So you put the plate on this magnet array. So when you put your magnetized cells in the well, they all come to the magnet. And then you take the magnet out. You only need 15 minutes on the magnet for most cell types. And they come together. You take out, put it in the incubator, and the cells generate a spheroid. Some cells, some applications, we leave on the magnet longer, but you don't have to. So, and of course, when you want to manipulate, you can put it back on the magnet. So this is the reproducibility of the technique. So this is a 384 well. This is just HepG2, liver carcinoma, and it's a 600 cells all the way to 20,000 cells. You can see very reproducible and very scalable. We published this little application of the Promega Pub Hub with Promega, where we show here, this is number of cells, this is diameter, the error bar very tight, and this is number of cells, and cell titer glow response very linear across the different range of cell numbers. So it's a very reproducible technique. Of course, this is, it's always great to see function, and that's one question that we always get when we try to publish, when we're publishing, is show us function, show us function. And this actually, I don't have this because this is the next generation of this work with cardiomyocytes, but our first paper was actually with Dr. Akei's department using cardiomyocytes, Dr. Ravi Bila. And so we published that, and now we're using IPS, human IPS cells, and this is 1,000 cells in a micro-L, and you can see, let me see. Why is it not beating? There we go. See? Can you see the beating? It's always very nice. And they beat for, as you see, we got them beating very regularly for a week. We have done the electrophysiology, and actually we had some very cool results. Yeah, it's beating. Yeah. They beat it beautifully, and we can control that. Actually, this is a work done by AstraZeneca. It was independent of us. Actually, the AstraZeneca guy sent me in an email this poster that they bought the kit, and they adapted the kit actually to do electrophysiology with cardiomyocytes. 3D culture is not trivial to do electrophysiology because it's not the same as, you know, patch clamping. It's not going to work. It's very hard. So because physically you need a stable system. You need things to be held together in place without moving. And that's what they did. They actually, because we can use magnetic field outside of the wells, they generate the spheroids. They generate the spheroids, which are magnetized, of course, and they place the magnets on top of the electrode, and they were able to hold them down in place and actually get very nice, this field potential, this impedance. This is the magnetized culture. This is the magnetized culture. This is non-magnetized culture that they tried. So again, it's enabling technology. It's not doing exactly what the technique was intended to do, but because the culture is magnetized, now you can come from the outside and hold them in place, which is not trivial. This is following up on the T cells. And actually, this is not quite Jim Allison's lab. It's right next door to Jim Allison. Jim Allison is the MD Anderson person that got the Nobel Prize for T cells 2 days ago. So, and actually he will be on this project on the second phase. This is a phase 1 grant that we got from NIH with Dr. Cooper's lab. And, and this is, again, it's a proof of principle taking advantage of magnetized these cells and being able to manipulate these cells with magnetic fields. So what we did here, we just took leukemia cells, which are not cells that grow in 3D. But we want to manipulate them, and we want an easier way to see how T cells interact with the cancer cells. So we took CD19 leukemia cells, CD19 positive, which are red cells, and CD19 negative, negative leukemia cells as well, which are green. So we culture them separately, magnetize them separately, and then we mix them together. The T cells are not magnetized, but we want to see if we can determine the targeting. So if you look at the image here, it's a little bit, yes? These are cell lines. These are cell lines. This is a proof of principle. We're doing that next, but these are cell lines. It's just proof of principle. So you cannot see very well here, but on my screen, it looks well. So So we have so we took the two CD19 positive, CD19, mixed them together, okay? So there's red here. It's hard for you to see on here, but if you want to see it, it's on my screen. You can see the red. Then we add the T cells. The T cells target CD19 positive cells. On this, there's no red after 48 hours. So they killed, and we could very easily detect that and quantify because of the magnetic field. We can concentrate them right on the field of view, and now we're going using solid tumors, and the next goal it is to use patient cells. Yeah, so these we just bring them together, yeah. So now going to cancer, and as I say, when we first started doing 3D cell culture, again, I was not a biologist. People said that this would never work, and there was a lot of hand waving as far as in vivo similar to 3D cell culture. So this, and we have enough data, not this, but this is a really important work that just came out this year, where we actually took a biopsy sample from the patient, we put it in the mouse, we created the PDX, and then we took the PDX and then we magnetized the cultures, generated the cultures, and compared the PDX to the culture. So here are the results. Here's the work of Dr. Jeff Bartholomew at MD Anderson. So with us. So here we magnetized the cells, took the tumor, we broke it apart, we used the magnet to bring them together. And one thing that is important here, he did not want to use our system, which is fine. So until he did all his work using the gravity techniques, round bottom, micro pattern plates, and then he did all the optimization with the cell lines. Fine. He went to the pressure samples, which are the PDX samples, which take forever to grow, and he tried to do the cultures. He could not get reproducibility from the gravity plates using the triple negative breast cancer cells that he had. So I was visiting MD Anderson, and I bumped into him, and he's like, Glauco, I cannot keep the cells together. Because he wanted to really bring them together and try to keep the heterogeneity of the system as much as possible. So I was like, Jeff, let's try. We'll come up with a protocol for you to magnetize your tissue quick, and we use the magnetic field to keep them together. That's what we're good at. We're good at bringing cells together. So it's like, okay, we're trying. So he tried, and as I mentioned to you, we usually leave on the magnet for 15 minutes, right? So he left on the 15 minutes. He called me, he's like, Glauco, works great. They're all together, let's see. 2 days later, I called him, and he's like, it didn't work. He broke it apart. The cells died. So my question was, did the tissue break apart and the cells died? Or the cells died, then the tissue broke apart? So to determine that, I told him, don't take off the magnet. Let's leave it on the magnet now. Let's see. And that was the difference. Pushing the cells tight together with the magnetic field made the difference. That's getting, just every time we do it, he gets it. So this is the result. So this is a case that we leave the cells on the magnet. So he took the tumor, broke it apart, did the traditional technique that he's doing. We took the other half. We came up with a protocol that we break the tissue apart, we magnetize, and reassemble. And these are the results. So this is, he looked at Icadira, Environmentin, PISMAD, KI67 for viability, and this is zoomed in. So this is the tumor, and these are two separate cultures. And you can see the morphology. And we did gene expression as well. It's the same, which also shows that the nanoparticles don't do anything to the cells. So this is, it's the kind of data that really gets us excited, and we're on the right direction. So, and he cannot grow this in 2D. They don't even grow. It's not even a question if it's different in 2D. He cannot keep these cells on growing. And when you're taking cells from in vivo, you have different populations. And if you go in 2D, you end up with fibroblasts. It's very hard to keep the different populations. So in this actually, we're going to patient samples now. This is a bone biopsy, small bone biopsy from prostate cancer. We broke it apart, we magnetized. We actually got 96 hours after, we got a very nice response for doxorubicin, which is the line of treatment. We've done about 20 patients, but again, handling biopsy and patient samples is tricky. You really have to, the manipulation and how you collect the cells is very important. So this is going through the high throughput screening aspect of technology. This is a work funded by NIH with the Scripps Research Institute and Cold Spring Harbor. It's a grant that we, 3 of those got it. And now we're moving to really high throughput tools. This is a 1536 plate. And again, when you add the cells on the well, the cells all come together. And if you don't have the magnet, they all dispersed. So these are, my fault, sorry. So this is just a picture of the plates. And again, the beauty of the yes? Yeah, it's a good question. And that's where we started. We didn't want to do any of this when we first started academically. We wanted to do a microchip to do neuron patterning, look at mechanosensitive cells. You can do it, but technologically it's tough because of heat. You need a lot of current. And those things heat up I have a picture of frying the cells, literally. So that's the challenge. But there are ways to cool it down. Technologically for us, it was just easier to use the permanent magnets. They're strong enough. But you could, yeah. Yeah, you can. There's no reason why you can't. Yeah. Especially when you go to microfluidics, smaller space, that's probably the direction we go. But that's where we started. I can tell you a story how this really started. They had nothing to do with magnetic levitation. We did that experiment. We burned the cells. It worked. And then I just put magnets on top of the petri dish. I had the magnetized cells. The next day I came, I looked at the cells. They're actually levitated. And I'm like, I can get a Nature paper on this. And we did. 90% of the biologists, I was in a big group. They never believed that we would make it work, but it works. But we started with yeah. So this is the reproducibility of the spheroids, and we just published this year, and I'm just going to give you a taste of the results. So here we took pancreatic cancer cells from 2 patients. Age 1 is 1 patient, age 1 is another patient. So within the tumor, you have your fibroblast, cancer-associated fibroblast. So we separate the cancer cells from the CAFs, which is the cancer-associated fibroblast, and we test 2D versus 3D in high throughput. And this is the library we used is FDA library, 3,000 components which are all FDA approved. So we're just doing what is the difference between 2D and 3D. And it was interesting, 3D always showed less hits, which in some ways you're eliminating the false positives, which the false negatives. And the false negatives is what it really makes drug development expensive. So if you can remove the drugs that don't work, that's great. So zooming in the results a little further. So this is just showing 2D activity, 3D activity. So every dot here is a drug. Whatever hits the white, it didn't do anything in 2D and 3D. Again, these are aspirin, Tylenol, all FDA approved drugs. So most of them didn't do anything. The pink are 2D active only. The green is the overlap. You go on the 3D only, we pick this one here, the most active, which is disulfiram. And disulfiram is in phase 1 clinical trial for pancreatic cancer now. So it's being repurposed. So we kind of picked the needle in the haystack, which is, we're pretty excited about it. Now we're digging more. We just finished 150,000 compound screening now. Nobody has done this big of a screening yet. This is going to be published pretty soon. But again, out of the 150,000 components, do you know how many we picked? 730. So that's why people use these huge compound libraries, because it's a very small percentage. But the ones that you get in 2D that are false negatives makes it very expensive. So I'm looking at my time here. So I'm going to, because I'm going to change gears quickly here. which is the 4D that we touched. We're talking about 4D in a lead dinner yesterday, and I'll go quick here because I'm not sure how I am on time. Oh, I have 5 minutes, plenty of time. Okay. So you magnetize the cells, you can bring the cells together fast, right? And that's great. And then we start looking at the cells when, within hours of bringing them together. And we start seeing that there's a lot of motion. And usually the cells actually, the culture contracts, they shrink first. So our idea was like, okay, why don't we look, can we use this process as a readout for biology? So that's sort of the hypothesis. If you have real-time imaging, can we, watch this process to look at drug toxicity or drug interaction? And that was the sort of the hypothesis we have. So the first application we developed was actually with AstraZeneca, and it was one of those things that I know I could print different shapes. We're able to print rings for wound healing assays, and I talked to a friend at AstraZeneca, and it's like, I'm wasting my life doing what they call aortic ring assay, which is a vasoactivity test, where they take the rat's aorta and basically they stretch and see. If you have a vasoconstricting drug, which may increase your blood pressure, it's tighter. If you have a vasodilating drug, it's looser. But it's not reproducible, it's not high throughput. It's very hard, but that's the way they test vasoactivity. So we came up with the hypothesis of what about we take vascular smooth muscle at first, we get a ring magnet, and print a ring, and see if we can do that. And that's what you're going to see here. So here, remember, I told you when we bring the cells together, they tend to contract, right? So these are rings of vascular smooth muscle. And we use blood. You cannot see the ring here because of the resolution, but there are rings. And we use blebbistatin. Blebbistatin blocks phosphorylation myosin light chain, so it's a vasodilator. So you're going to have high concentration of blebbistatin. This is the control. Remember, they're going to contract. If you're using a vasodilator, we're hoping this contraction decreases as a function of concentration. So look at that. So control. This is overnight, so this is fast. And now you can see the ring better, see? You can measure. This is just the size, and we develop a software to do the image analysis, of course, which pixel count is pretty straightforward, and you can quantify the process. So this is time, and this is the change in size. But the shape of these curves, there's a lot of biology there. We're fingerprinting, as I'll show in a few slides, fingerprinting the cell types. But you remember you saw an iPod next to the first image? Just like a phone, that we use the iPod. All the measurements are done with an iPod because we develop an app, and we can measure this very easily. So, and of course, the biology. Blebbistatin blocks phosphorylation myosin light chain. This is your control. You add blebbistatin, and you can clearly measure the control. So the biology is there. We compared actually rat to human. We use vasodilators, vasoconstrictors, and we published this in scientific reports, and it works really well. We're actually using this, and we published as well with the myometrium smooth muscle, which is for preterm birth contraction. So we use the ring, then we figured out that we can use the dot too. There's a few reasons why. So we can print a dot, and this is, again, using this tool for vasoactivity. Again, these images are straight from my iPod. So what you see here are dots of cells. These are 3D cultures. This is a 3T3 fibroblast. This is retinoic acid. We're just using this as a measure for toxicity. And again, time is the time of the experiment here. If you watch this, so high concentration, see the cells contract in the first 3 hours. 3 hours you have your dose sensitivity for toxicity of retinoic acid. instead of taking 10 days, which is how long it takes for the gravity techniques looking at growth. We're just looking at this assembly process. And, but what is important is the shape of these curves. There's a lot of biology that people have not looked at it. And that's this slide. We published this. We used 6 drugs and the same cell type, same system. And these are just dose dependence curves. But the shape of these curves are very different. This is SDS, we just break. So we're trying to fingerprint mechanism of interaction. Again, you can think, you're putting the cells together, they're interacting, right? It's time-dependent process. And if the drug is interacting on the surface, on the cytoplasm, or the nucleus, it's different time points. And we're trying to pick up, or if it's a scaffolding reaction. So this is new, it's very new, and we're really, it's pretty powerful. So this is actually, because there's so much data, there is the shape of these curves, qualitatively you can see it. So we're using a principal component analysis, which is, you know, it's a multidimensionality reduction tool to fingerprint the cells. So here are pancreatic cancer cells and normal cells. So when you do the PCA, based on modeling those curves, and if you want to talk about that, I can talk after the break, you can actually segregate. These are the cancer cells, these are the normal cells. So we're actually fingerprinting the different cell types, looking at the shape of these curves. And this is the system that we use to do all these measurements. This is in the incubator. It's just iPods. And if you think about it, that you're taking a picture of 3A and the 84 plate every second, there's no instrument that has that type of throughput than we have. But we're not looking at microscopy. We're looking at the culture. Why we came up with this? Because we're losing students taking pictures forever. And so we, like, instead of taking pictures, let's develop a new tool. And that's, you know, we developed out of paint. So we can print different things. These are just playing around with fluorescent cells. We actually generate organoids, and this was a work that just came out in biomaterials, which we took salivary glands, we took neuronal, actually, stem cells that differentiate into salivary glands, reimplanted in the mouse, and actually re-innervated the tissue, and there was function too. So this is pretty cool. It's great work of a colleague of us, a collaborator, Jean Ferreira. And I showed this picture because we were in the space station this year, early this year. This is Peg Whitson doing experiment with our system at the space station. And why, I don't know if you know, in space, cells grow in 3D. There's no gravity. So why do you need a 3D bioprinted 3D cell culture in space? It's very hard to manipulate cells in space. So for change media, it's not trivial at all. They do by dilution. So you have to carry a large volume with you to change, to fix your cells. So we send magnetized cells so we can use the magnetic field to hold them down and you can change the media. And nobody has done the 2D control in space. They have tried. So we send magnetized cells to try to print them on the surface. Actually, we did print them on the surface, hoping they would spread and we'd get very clear results. It's not. The gravity has more to do than, it's not contact, because they stick, but they don't spread. Again, the problem with space is that there's a lot of work to do it. The experiments are very tight. So these are our collaborators. I'll be glad to talk to anybody, answer any questions afterwards. Thank you very much.